Thank you very much, Ludmilla, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, thank you so much. I'm happy to be here. And let's see, I'm very excited to share some of the things that we've been doing in my group for the past few years. And let me see if I can make everyone smaller so that I can see the slides. There we go. And before I go into what my group is about, I just wanted to kind of say a little bit about me and my training. I got my PhD from UC Berkeley before doing a stint in Europe, specifically in France, as a Fulbright scholar after my PhD, and then went to Princeton for my postdoc, and that's where I learned all the metabolomics that I do now. And half of my lab, we still do quite a bit of surface chemistry, which was my the focus of my work when I was at Berkeley. And then I started my group actually at Cornell University. I was there for 7 years. I got tenure, and then Northwestern came knocking and convinced me to leave Cornell and go to Northwestern. And obviously they were successful because I'm now at Northwestern. I've been there now for 4 years. So this is my 11th year as a faculty, with 7 years at Cornell and now 4 years at Northwestern. And the purpose of my talk today, I kind of tailored it based on kind of my conversations with Kate, and what I think this audience will be interested in is looking at leveraging metabolomics, which is the expertise of my group on the bio side of what we do, to explore the networks of reactions that are involved in carbon metabolism, in carbon cycling, but more importantly, we do this in bacteria that have biotechnological relevance. And just a little bit of my group, we are just excited, every member of my group, about unraveling mechanisms of organic processes. And the purpose of this is that we want to get new insights at the interface of either environmental biochemistry, environmental chemistry, and environmental technology or environmental biotechnology. And this has implications for environmental processes, environmental sustainability, agricultural productivity, ecosystem health, nutrient cycling, carbon cycling that you can leverage for engineered biotechnology or recycling. But what we get focused on the mechanisms, and my colleagues and my peers can use that information for engineering purposes. And I just wanted to give you kind of this was the most recent picture as of 2 months ago. We had a couple of new people join. But that's the most recent picture we had for now. And I just wanted to share that we are just a group of researchers who get excited about chemistry and biochemistry of organics in environmental processes, in environment biotechnology, as I mentioned before, if within the context of either bioreactors or wastewater treatment or agriculture. We are intellectually diverse, and we're also quite ethnically diverse. I myself am from the Caribbean originally before moving to the States some 26 years ago now as a little girl. We also have people from the US, of course, but from Central America, Africa, and the Middle East. And I'm very proud to having such a diverse community of students and scholars in my group. And our motivations, specifically within the context of looking at biological organic processes, is to understand the biochemistry of these processes, especially within the context of natural carbon cycling, and how that can be linked potentially to innovations of engineered carbon cycling. And our central research question is, what are the metabolic mechanisms that control cellular carbon conversion in soil bacteria of biotechnological importance? And the reason that this is important, not just only within the context of environmental carbon cycling, which is tightly linked to projected climate change and what's going to happen to our global carbon budget. We have an interest in this, but also these mechanisms are important for designing a suitable microbial platform for a specific target objective. And this target objective could potentially be for waste conversion or product biosynthesis, all of which requires a fundamental understanding of the native metabolic regulations in order to either exploit, rewire, or supplement the native metabolic reactions. And the way we do this in my group, our central kind of expertise is metabolomics. And in a nutshell, metabolomics is the high resolution chemical fingerprinting of carbon metabolism. For central carbon metabolism, we've developed methods that can look at up to 200 to 300 metabolites simultaneously. And we can use the levels of the metabolites and dynamics in these metabolites to track what is happening in the network of metabolic reactions. And the instrument that we use, we have two high-res mass spec. This is one of them. Mass spectrometry that we use in the lab with Orbitrap technology. And so this is an auto sampler, just to give you a sense of how these instruments look like. And this is the injection syringe that take, auto sampling. We can have about 60 to 80 samples at a time. It goes first to UHPLC for liquid chromatography for separation, initial separation of the different compounds, and then it goes into electrospray ionization and eventually to our Orbitrap mass spec. And the advent of LC-MS, specifically high-resolution LC-MS, has been quite revolutionary when it comes to the omics technique, specifically metabolomics, because it allows us to be able to get at information we couldn't get before. So back in the day, people would only use an LC, so use the retention time in the LC column, a liquid chromatography column, to separate different compounds. So you can see here, this is fructose 6-phosphate, this is glucose 6-phosphate. If you only have LC, you can separate those two. But if you look at here, ribose 5-phosphate and xylulose 5-phosphate, they will co-elute, which means that they will come out at the same time in the LC column. But the beauty of having a mass spec coupled with the LC, you can separate these different compounds based on different M/Z channel at high resolution. So we have high accuracy up to the 4th decimal point in the mass over charge value that allows us to be able to not only get different compounds that have different mass of M/Z, but same retention time, but also get at compounds that have the same M/Z, but come out at different time elution time from the LC column. So using this is how we're able to develop methods that can look at multiple compounds at once. So in my lab, there are multiple ways that we leverage metabolomics approaches. The kind of basic way we use metabolomics is to profile the metabolome. I'm just showing you just a snapshot, a short version of it. This actually goes very long. This is looking at Pseudomonas putida grown on 2 different substrates. And you can see just from looking at the profiling of metabolites that you have some compounds, metabolites that are depleted in the presence of succinate, which is a gluconeogenic substrate, as a substrate that feeds into the TCA cycle. And then if you're grown on gluconate, which is a glycolytic substrate, you have elevation of this metabolite. So the metabolome profiling can give you quite a good sense of which reactions are actually upregulated or which reactions are more operational, are more active relative to others. That's our basic way we use metabolomics. I would say 90% of our collaborators, that's all they need as information. But beyond that, where really is the power of what we do is to combine this with 13C or 50N with stable isotope tracers. And then with stable isotope tracers, there are 3 ways that we use metabolomics approaches. One way is to look at steady state. We will feed the cells with a 13C label substrate and look at where the labeling goes inside of the entire cellular network. The second way we do this, we can use fragmentation to look at specifically which atoms are labeled, and that allows us to do a mapping of the routing of carbon inside the cell. And the third way that we use this is to use 13C kinetics. And for 13C kinetics, I believe my lab is one of 3, maybe 4 labs in the world that do this, is to do very high resolution from seconds to minutes in tracking in vivo enzyme activity. And as far as I know, that's the only way you can do in vivo enzyme activity instead of doing in vitro enzyme activity that's done all the time. Okay, so this is kind of looking at metabolomics, but you might ask yourself, what is the connection of this to other omics techniques? And we combine metabolomics with fluxomics. So metabolomics, you get the metabolite pools or the tracing of stable isotope through the metabolite pools. Then we combine this with the phenotype of the cells to get that quantitation of metabolic fluxes. But this information is giving you phenotype information, but there's a lot of interest in connecting that to the genotype. So how is this connected to the other omics techniques? So we have the DNA sequence that give you the boundary set of what may happen in terms of metabolic reactions that may happen in the cell. And from this, the next higher resolution is transcriptomics that tells you, okay, a subset of the genes can then now be transcribed, and that you go from the boundary set of what may happen to now what can happen in the cell. And proteomics gives you the abundance of proteins. So you go from from the gene expression to what can happen. A subset of that can be translated into proteins. So you go from what can happen to what should happen in the cell. And metabolomics is telling you actually what is happening in the cell, because now you can follow the substrates and the products. And the fluxomics is telling you the rates at which they happen. So we use our expertise in metabolomics and fluxomics to inform to what extent the other omics techniques are predicting what is happening. Okay, so what is the kind of insight or the reason why we would want to combine different omics techniques, which is what we do? It's because by combining them and seeing discrepancy between these techniques or agreements between these techniques, we can get information about regulation. So if you combine genomics and transcriptomics, you can get information about transcriptional regulation. Combining transcriptomics and proteomics, you can get information about translational regulation. Combining proteomics and metabolomics, you get information about post-translational regulation, and combine metabolomics and fluxomics, you get information about feedback regulation. So in my group, what we try to do is we put a lot of effort in applying a multi-omics approach to really uncover the regulation of the network of metabolic reactions for the purpose of guiding metabolic engineering outcomes, or for the purpose of predicting what could happen in an environmental setting. So the first part that I'm going to cover, I have two vignettes in our kind of work that we do, and I leave quite a bit of time for questions and discussion. The first vignette is looking at the metabolic network of Pseudomonas putida. We've done quite a bit of work on Pseudomonas putida. And this work, if you want to look at it, some of it was published in Journal of Biological Chemistry in 2019. And another work that's looking at how nutrients can actually remodel carbon metabolism was published in PNAS in 2020. And these are the people who were mostly involved in this work. Matt is actually currently a PhD student at UC Berkeley, and Carol is now working as a biotech engineer in Boston. She was a PhD student, graduated a year and a half ago. Rebecca is now a postdoc now with Greg Beckham at NREL in the DOE lab. She graduated now 2.5 years ago. And Mina is now working as an enzyme engineer in San Diego, and she graduated a year and a half ago, and they were all involved in this work. And these are the two current members in the lab, or current postdocs, Nenching and Pendishwari, were involved in this work. Okay, so the first thing we looked at here is looking at, we were very interested in mixtures because Pseudomonas has very nice capabilities of using multiple type of substrates, and Pseudomonas is one of the main workhorse in metabolic engineering all over the world, specifically quite so in the DOE world. And we wanted to understand how is it that they can use sugars like glucose, that E. coli is well known to use sugar really well. E. coli is one of the premier model organisms, but E. coli cannot use an aromatic compound, but Pseudomonas can. So we really wanted to understand the network underlying that. And first, we did some basic growth phenotypes. So we grow Pseudomonas putida and carbon equivalent amount of glucose. So here we grow them on 100 millimolar, 50 millimolar of glucose. And here we grow them on the exact amount of the same carbon, but 1-to-1 ratio of glucose and benzoate. And we see the rate that they're using this. If you sum the rates that they're using, the benzoate and the glucose here in the mixture is the same as the glucose by itself. So that tells us that they can use both carbon type, both substrate type, simultaneously at the same rate as if they were fed on sugars. And if you look at the growth rates, there was no compromise in the growth rates. They have approximately the same growth rate. So then from this, we wanted to know, where does the carbon go inside the cell? This tells us they have the same consumption rate. Do they distribute the carbon everywhere equally inside the cell? And the way we probe this is by growing the cells on 13C labeled glucose. So the glucose carbon was fully labeled and a non-labeled benzoate. So the data we're going to show, if you see non-labeled fractions, you know that's 100% from benzoate. If you see a fully labeled fraction in the metabolite, you know that's 100% from 13C glucose. If there's any partially labeled, then you know that there are carbons that are sourced from both glucose and benzoate. The other thing to keep in mind, that just as a way to visualize and track things better, I color the atoms based on the pathways that the different atoms come from for making the metabolite. So if it's red, it's coming, that specific carbon was coming from the pentose phosphate pathway, which is a pathway that makes pentose sugars that goes into DNA backbone. If it's purple, then it's coming from the TCA cycle. If it's blue, it's coming from upper glycolysis. If it's light blue, it's coming from downstream metabolism. And if it's gray, it's other, like dissolved CO2, for example, that goes into the metabolite. Okay, so let's start somewhere. We start with nucleic acid. So this is a nucleic acid precursor. This is inosine monophosphate. This is the precursor to purine ribonucleotides. So we go them on glucose label, as I just mentioned, with the star, that means the glucose is labeled, with benzoate non-labeled. We also go them on glucose labeled by itself as a reference. And we can see if it's only glucose, it's mostly fully labeled. That makes sense because it's all coming from glucose for the most part. Some of them is partially labeled, and the reason for that is because these ribonucleotides are also made from dissolved CO2, which will be non-labeled. But then when you switch to the glucose label to benzoate non-labeled, we see about 20% is fully labeled. That means 20% of this metabolite is made exclusively from glucose. About 60% of it is made, or actually 70% of it is made from partially labeled of different isotopomers. And then 10% is all non-labeled. So here we already see that there is not equal partitioning of the carbons, because if there was, everything would be partially labeled. So we keep going. Let's look at a cell membrane precursor. This is N-acetylglucosamine. So for N-acetylglucosamine, we see in the glucose label, the same as here, it's mostly labeled. So that's our reference. And if we look at glucose benzoate, all the fractions are partially labeled. So it tells us this precursor is made from carbon from the sugar and also from the benzoate. Then when we get to amino acids, so protein precursor, that's when it gets super interesting. So we see now for valine, which is made from downstream of glycolysis or the Entner-Doudoroff pathway in Pseudomonas putida. If it's glucose by itself, it's all labeled as a reference. That makes sense. But if it's glucose labeled and benzoate not labeled, we get about 45% is fully labeled. That means exclusively made from the substrate, the sugar substrate. We get about 30, almost 40% that's partially labeled. And we get this about 10, 12% that is not labeled. Then if we look at TCA cycle derived amino acids, again, you can see the reference with glucose labeled, it's all labeled as expected. And with the glucose benzoate, we don't actually get barely anything that's glucose fully labeled. That means that most of the fraction is coming from either a combination of glucose and benzoate carbons, or it's coming from exclusively from benzoate. So this tells us there was non-equal partitioning of the substrate carbons within metabolism, even though we see equal consumption, as I showed in the previous slide. So we wanted to quantify this, and this was, this was alluded to maybe 3 decades ago, but in previous experiments that people did. And in these experiments was the first evidence to show that complete segregation in metabolism on 2 substrates that they're equally using. And to get into quantification, what we do in my group, we combine the high-resolution carbon mapping using 13C assisted metabolomics, analogous to what I just mentioned before, but we do it throughout the entire metabolic network. We use the GOF phenotype, the substrate consumption rate, and the metabolite secretion rates, and that's for mass balance. And we also use the biomass growth to get at the carbon fluxes. So first, I want to show you the carbon fluxes for glucose. Here, I'm showing you all the fluxes are in terms of millimole of this metabolite, the reactant per gram cell dry weight per hour. And the main thing I want to show you here is that if the cells only have glucose, this is Pseudomonas putida again, they're going on glucose, and the whole point of the cells is to drive glucose flux to the TCA cycle, because that's where you get most of the energy. You get some energy through glycolysis, but a lot of the energy you get through the TCA cycle. That's where you get your NADH, eventually ATP via oxidative phosphorylation. So you can, the thickness of the lines are telling you the magnitude of the fluxes. But now the cells are good on glucose and benzoate. This is the metabolic flux quantitation based on our optimizing our data. We see, remember, the cells were going an equivalent amount of carbon for each substrate. So if we were to turn this into carbon, because glucose had 6 carbons and benzoate has 7 carbons, this is essentially the same amount of consumption rate as I showed in the growth phenotype data 2 slides ago. But the main thing to see here is that the magnitude of glucose-derived fluxes that goes into the TCA cycle is half. And the reason it's half is because the benzoate carbon flux is going to the TCA cycle. And the other thing that we see is that a lot of the fluxes actually maintain in the fluxes that go into biomass. So all the red that you're seeing here and also here are referring to fluxes that go into biomass growth, and these are maintained. And the reason they're maintained is because the benzoate goes into the TCA cycle. Here we didn't have benzoate, so we can still maintain high flux to the TCA cycle because of benzoate going into the TCA cycle. If you remember the previous slide, I showed that the TCA cycle derived amino acids were almost exclusively made from benzoate, and some of them were just a smaller fraction were made from the combination of glucose and benzoate. And here, look at how the flux is going in the reverse direction. Remember here, the glucose-derived carbon is going from lower glycolysis to the TCA cycle, but now it's going the reverse direction to feed into all these nodes that go into the biomass. So what we see is the metabolic fluxes that feed into biomass group, they're conserved despite the segregation of substrate investment into the metabolic network. And what we wanted to know is, are these metabolic flux changes predictable from protein abundance changes? And the way we go into that is by combining the metabolic flux change, the change in metabolic flux from the glucose-benzoate mixture relative to the glucose alone, that's the ratio that we're showing here, to the protein abundance ratio. So if you see for initial glucose catabolism, if you're in the same quadrant, that means that a decrease in flux also was correlated to a decrease in protein abundance, which is great. It's not the same magnitude, which is expected. You're not going to have the same magnitude of change, but at least the changes are going in the same direction. The lower glycolysis, this is endodopa pathway, which is the another glycolytic pathway, that we also see a decrease in protein abundance. A small decrease in protein abundance can lead to very high decrease in flux changes. Again, we see it's in the same quadrant, even if it's not the same magnitude. Then when we go to upper glycolysis, we see that actually you can have no change in protein abundance, but there is a change, almost twofold change in flux changes, which means this is not protein regulated. We see in the pentose phosphate pathway, again, barely any changes in the protein because the protein abundance changes on the x-axis, but significant changes in flux in the fluxes. And then once we get to the TCA cycle, it's all over the place. Sometimes we see, for example, no change in protein, but almost fourfold increase in flux changes. Sometimes we see a negative change in the protein, but there's an increase in fluxes. It just tells you that this is not dependent on the protein. Protein is not dictating what's happening. But some of the proteins, there is positive correlation, like these proteins here. If you increase the protein abundance change in the mixture relative to the glucose alone, we also see a corresponding increase in the flux changes. In gluconeogenesis, we also see that there's discrepancy. You don't see what you should expect. And the reason for that, these directionalities in the fluxes are based on turbodynamics and not on the protein abundance changes. But that's not the only thing. First, we see a discrepancy, as I highlighted here, a discrepancy in the metabolic flux changes and protein changes. And I don't have time to go into this data, but if we look at the paper, we see that these changes were superseded by favorable thermodynamics due to the hierarchy in the metabolism. So the next thing we look into, to kind of wrap up this section, we also look at, okay, now that we see this favorable thermodynamics, it's based on metabolite levels, different changes in metabolite levels. But so this is, if you look at the metabolite levels based on the protein abundant changes I just showed in the previous slide, we see that these reactions that involve these metabolites were regulated on the metabolite level. We saw that the reactions that involve upper glycolysis here were protein level regulated. These reactions are involving lower glycolysis, again, were metabolite level regulated. And then the reactions are involved in the TCA cycle were both metabolite level regulated and protein level regulated, as I showed before. And the other thing that we saw is that phosphorylation of the proteins, phosphorylation can actually turn on or turn off the protein. So we also look at phosphorylation, and we see that the reaction from pyruvate to acetyl-CoA was regulated by phosphorylation. To promote citrate going to alpha-ketoglutarate in the TCA cycle, we also saw that was promoted by, regulated by phosphorylation of the protein. So in addition to protein abundance changes, phosphorylation can also regulate the flux, so turn on or turn off to what level this protein is going to operate. And we also see gluconeogenic flux from pyruvate to phosphoenol pyruvate were also regulated by phosphorylation of the protein. This is why when I showed you the correlation graphs, there was no correlation between the flux and the protein abundance. So what have we learned thus far on the metabolic network of Pseudomonas butida? We see the metabolic segregation of carbons during co-utilization of carbohydrates in an aromatic substrate, which is gluconeogenic substrate. We see metabolic flux changes are only explained partially by protein abundance changes. So you can imagine if you were basing this on genome, there will be even worse correlation. And then the favorable fluxes were facilitated by metabolite levels, but also post-translational regulation. So as a map to summarize what I just said, this is where we saw based on the metabolite labeling, when the glucose goes in, even though we see equal consumption, a lot of it gets populated in the upper glycolysis and the pentose phosphate pathway. The benzoate is what dominating the flux of carbon into the TCA cycle. And these we see labeling coming from carbon sourced from both substrates in these kind of between the lower glycolysis and the TCA cycle. And the squares are showing you the different points that are regulated at the protein level. And the circle is showing you things that were turned on or turned off based on post-translational modification, also known as the phosphorylation of the proteins that I highlighted before. So the next part that I have in the next probably around 15 minutes or so is the second vignette, which is looking at bacterial conversion of carbons, not just from lignin, but also plastics. We've been working with kind of added to the list of bacterial species we're working on. We've been working a lot on Pseudomonas butida. Now we're working quite a bit on Chromomonas testosterone. And this work was published last year in Nature Chemical Bio. And the first author of this work was Rebecca that I highlighted before. And she actually stayed a year as a postdoc before she moved on to Greg Beckham's group. And we have our collaborator, Jake from University of Chicago, Adam Goss from the Oak Ridge National Lab, who does mutants for us, and Manu Neto Dominguez, who is from Pablo Nickel's lab from Denmark. And these are the other people in the lab who are still currently working on trying to understand the conversion network of carbon metabolism that is involved in the bacterial conversion of lignin and plastic-derived carbons. And the work that we've done has focused on Chromomonas testosterone. Chromomonas testosterone was actually isolated from a wastewater sludge. And it was initially isolated because of the fact that it can grow on surfactants, aromatic surfactants. And then from there, people have speculated, and there have been some preliminary evidence that it could use some other aromatics. And in our paper, we were focusing on the potential of Chromomonas testosterone to use vanillate and 4-hydroxybenzoate, which are two lignin derivatives, as well as terephthalate, which is a monomer related to polyethylene terephthalate or PET plastics. And I wanted to highlight here, there are 3 different paths that the aromatic carbons can take to eventually lead to central carbon metabolism. There's a 2,3 meta cleavage pathway, which is through here, that eventually gives you acetyl-CoA and succinate. There is the, this is the 2,3 meta cleavage. This is the auto pathway. The 2,3 meta cleavage gives you acetyl-CoA and pyruvate. And the 4,5 meta cleavage pathway eventually give you oxaloacetate and pyruvate. So first, we wanted to know which of these pathways are involved, because as you can see, these pathways have different energetics. This one gives you an NADH, this one gives you an NADPH, and this one doesn't. So then the way we first put this, before I go into our multi-omics data, I wanted to highlight that the information that we get from the mechanisms, information that we get from this work is not only for these 3 compounds that we look at, but they're also relevant for hydroxybenzoate, methoxybenzoate, and phthalates, which are just by diversity of other aromatic compounds that we are interested in just in general in the waste carbon conversion community. Okay, so then what we did a combination of transcriptomics and proteomics. We did the transcriptomics here at Northwestern. The proteomics were done by our collaborator, Jake Walbauer at University of Chicago. And we did this, so this is a lot of data. Let's see if I can carry you through this. These are the 3 different substrates. This is the 2 lignin derivatives, and this is the plastic derivatives. On the left column for each, the left side of each column is the transcriptomics data. On the right side of each column is the proteomics data. And these are separated in terms of the different cleavage pathways. So these are the results for the 2,3 meta cleavage pathway. The different enzymes are involved. This is for the auto cleavage pathway. And these are the proteins that are involved in the 4,5 meta cleavage pathway. And the thing I wanted to highlight here, these data are looking at the aromatic. This is a ratio of transcripts or protein abundance for the aromatic when the cells were grown on the aromatic substrate of either of these 3, relative to if they were grown on succinate. We use succinate as a reference because succinate is an intermediate. It's a short-chain carboxylic acid that goes into the TCA cycle, and they also grow well on succinate. What I want to point out here is that anytime you see a dark brown, that means that we only see the proteins only in the presence of the aromatics, that these proteins were not expressed. There was no abundance of these proteins. They were expressed, but there was no translation, I should say, because you can see the transcriptomics data is showing you high expression of these transcripts, but there's no translation into proteins. And that's very important. This is why I went to the whole purpose of going into highlighting transcriptomics and proteomics data. And so we know these are, at least some of these are transcriptionally regulated, and I'm gonna get into protein level regulation later. So first we see that there's no change versus the aromatic and the succinate. So the gray means there's no change in a lot of the transcripts that are involved in the autocleavage and the 2,3 meta pathway. Some of them, we don't even see the transcript at all. So it's very clear that this pathway is not very active in the aromatics. And then the proteins, no proteins are abundant at all. Then we get to the 4,5 meta cleavage pathway. It's very clear. You're getting almost 300-fold increase in the transcripts. And then we see a lot of the proteins, select proteins are only, not only there's a high increase, but some of them only in the presence of the aromatics. So this transcriptomics and proteomics data confirmed only the 4,5 meta cleavage pathway is actually active, even if the genes, the genome of this bacterium is showing that they have genes that are involved in the other cleavage pathways. This is where you can have discrepancy. You can have the genes. It doesn't mean that they're actually operational. So then the next thing we did was to look at to what extent they grow on this different carbon source. We have the succinate here again as a reference, and we have the 2 substrates are related to lignin, and this is a substrate related to plastics. As you can see here, they grow really well on the substrates, which is good news for us. But the one thing I want to highlight is the 4-hydroxybenzoate is the one that you have quite a bit of the carbon is actually lost via secretions. And that's not what we want. We want to have very high yield of the carbon either towards biomass or towards a product that we want to make. So anytime you see metabolite secretions in bacterial work, it's a good indication that you have bottleneck going on in the metabolic reaction, which then induce secretion because of the bottleneck. So we wanted to identify where these bottlenecks are. And the way we did this is by systematically really measure the secretions of metabolites that are in solution by using exometabolomics. Essentially, it's doing metabolomics profiling as a function of time for the extracellular medium. And we do kinetics on this in order to be able to get rates. So I wanted to highlight here, not only we're seeing metabolites throughout the 4,5 meta cleavage pathway. This is another confirmation. This is a pathway that is operational because we see PCA here, which is from this pathway. We see PDC here, which is also from this pathway that is being secreted. We also see secretions of pyruvate that is later in this pathway. And this is the rates of secretions I wanted to show you here. So the highest secreted metabolites were coming from the 4,5 meta cleavage pathway, which are the two gray here. Some from the TCA cycle, the alpha ketoglutarate, and then a few from lower glycolysis. One thing I wanted to highlight here, which was, something we stumbled upon, that there's actually secreting a metabolite that is a valuable compound. I know Greg Beckham's group has actually engineered bacterium to try to get at high secretion. They've engineered Pseudomonas petita to get a high secretion of this metabolite that is a valuable precursor to polyesters. Okay, so now we see that we identified the bottlenecks. So then we wanted to know, where are these bottlenecks? And the way we further go into this, and I'm just going to show you one node that we studied quite a bit, is the oxaloacetate node. And the reason this node is so important in metabolism because the oxaloacetate is between three different places. It's the node that controls three different routes that can happen to the aromatic carbon. Either it can go to the oxidative side of the TCA cycle. So that's one fate of that carbon once it gets to oxaloacetate. It could go to the reductive side of the TCA cycle. That's a second fate potentially of this carbon. Or it could feed it into pyruvate, which will be cataplerosis or gluconeogenesis. So we use our labeling experiments. So in this case, we are feeding the cells labeled for hydroxybenzoate, partially labeled for hydroxybenzoate, and we use MS-MS fragmentation to look at not just the fraction of labeling, but which specific atom is actually labeled. And from this, we can see that actually looking at the phosphoenol pyruvate, which is here, we see that this fraction, about 20% of it is coming from oxaloacetate and 80% of it is coming from pyruvate. So that allows us to get some insights into, after the 4,5 meta cleavage pathway, how things are regulated at the oxaloacetate node. So instead of going to, essentially, if you can get things to the phosphoenol pyruvate, you can either get it directly from oxaloacetate or pyruvate gets fed into phosphoenol pyruvate. And these things have different energetics consequences. So speaking of energetic consequences, we also look at carbon cycling, carbon fluxes in terms of the cycling of this carbon through the different pathways. And here, this is the kinetic isotope flux profiling that I mentioned before. That there are only 3 labs that do this because this is seconds. This is 120 seconds looking at labeling of substrates into different metabolites. The key thing I want to show you here, I just mentioned how oxaloacetate is a node of regulation. You can see if we had the cells on 13C labeled succinate and we switched them to non-labeled version of the 3 aromatics and succinate as a reference. And we see just within 120 seconds, they're labeling really fast. But then once we get to phosphoenol pyruvate, we see only the succinate, but we don't see the aromatic, the derived carbon. So that tells us there was a bottleneck in the production of PEP from pyruvate, even though the MS fragmentation tells us most of it comes from pyruvate, but there's a bottleneck there. And that is the reason why we had this high secretion of pyruvate in our metabolite secretions. So as I mentioned before, if you have high secretion of a metabolite, it's an indication that you have a bottleneck. And indeed, we did see a bottleneck. in our 13C metabolomics data. So then the next thing we do, analogous to what we've done before, we compile all these 13C metabolomics data and with our GOF phenotypes to then try to understand what's happening to this aromatic carbon and the flux of what's happening. And this is, the colors are telling you increase in fluxes in 4-hydroxybenzoate, which is one of the lignin derivatives, relative to succinate. Again, succinate is used here as a reference. And we see, for example, if the cells are going on the aromatics, they have much less flux. You see a decrease here. That's what the dark blue is saying. There's the decrease in the flux from succinate to malate, because of the fact that the aromatics do not need this high flux from succinate. What they actually need is the cells, when they're going on the aromatic, they need a high flux coming from the carbon to go into the pyruvate and the oxaloacetate and go to the oxidative side of the TCA cycle. So it really helps us to put some quantitative significance into the cells processing one type of carbon versus another type of carbon. And using that high resolution metabolic flux that I show here, we can actually get at the energetics, which are really hard to measure just intracellularly, but because we can get high resolution of our flux analysis, we can get at this information. And what I wanted to highlight here is the fact that the 4-hydroxy benzoate uptake, it, even on one hand, it consumes NADPH, but in the 4,5-meta cleavage pathway, every NADPH that it consumed, it actually produces it. So it's a net balance in terms of NADPH, which is fantastic. And that's, we think, one of the reasons why the cells prefer to use the 4,5-meta cleavage pathway instead of the other cleavage pathways, because of this generation of NADPH in that pathway. The other thing I want to point out is catapleuroses is a major source of NADPH for these cells, which is not the case for succinate here. So on the left is 4-hydroxybenzoate, energetics, and on the right is succinate. And the last thing I wanted to point out is the dark gray here is transhydrogenase. Because of the surplus of NADH here, the cells use transhydrogenase to turn this NADH into NADPH, to have NADPH for the cells. But then on the flip side, we see here, they consume the NADH here and turn it into NADPH here. So in the aromatic sense, we see that we have an extra like NADPH consumption that can be made. We have a surplus in NADPH that can be then translated into NADH. In the succinate side, we have actually a surplus of NADH that can be converted into NADPH. So different energetics for the different carbon types. So what is kind of the insights that we got from this multi-omics investigation of lignin plastics carbon derivatives by Komomonas testosterone? We see the aromatic substrate uptake is transcriptionally regulated. We see the central carbon metabolism is not driven by changes of transcripts or protein levels, except that protein level regulation at one node, which is the oxaloacetate node, that most of the fluxes in central carbon metabolism are thermodynamically regulated. And this is very important for, the audience here who's interested in looking at thermodynamic regulation and to what extent that to build on this to do kind of designing a cell, or if you wanted to kind of manipulate a cell. So what are the trends? You can do genome manipulation and you might not get a phenotype because of thermodynamic regulation. And the bottlenecks, so we work with Adam Gus to resolve some of the bottlenecks that I mentioned before. And what we did, the overexpression of the PCA, remember we had secretion of PCA, so that's a bottleneck. We also, he also prepared overexpression for the PDC conversion into the downstream metabolite. That was another point of bottleneck, or just overexpressing the entire operand. And based on these results, we saw like, so the dotted line is the wild type, and these are the results from the different mutants. Then we can see that for if you overexpress the PDC, the enzyme that convert PDC to the downstream metabolite, or the entire operant, we don't see there's no significant change relative to the wild type. The only thing that really increase the rate of consumption of 4-hydroxybenzoate is this overexpression here. And then the next thing we look at the PCA secretion. Remember, I told you this is the level of secretion we saw in the wild type. And lo and behold, by overexpressing this enzyme, we get the secretion is drastically decreased, which is the goal. However, if we overexpress this, we see that the secretion is actually increased. So we really need to overexpress either this enzyme or the entire operant to relieve the bottleneck. The next thing we look at is the PDC levels. And if your interest is actually to make PDC, which I say is a valuable precursor to polyester, by overexpressing this enzyme, it moves the carbon flux to PDC, but then there's still a bottleneck. And so that you see this huge increase. This is a level of PDC secretion we had in the wild type. And we have 1 order of magnitude, almost 2 orders of magnitude increase, almost 100-fold increase in the secretion. And if that's your target valuable product you want to make, and that's a good thing that you want to do that. And then if you overexpress, of course, this enzyme that is downstream of PDC, we get completely, the secretion is about the same as the wild type. And the entire operand just drives all the carbon flux towards central carbon metabolism. You get the same result here. And finally, we measure the pyruvate secretion. This is a pyruvate secretion that we got in the wild type. And we see every single changes that we did in the cells increase the pyruvate. And the reason for that is that you're driving so much carbon flux towards pyruvate. Now there's a bottleneck at pyruvate, and then that leads to secretion of the pyruvate. And just so some things that we're looking forward into doing in my group is looking at the mechanisms of mixed substrate metabolism. We had some preliminary data of this in this paper, and we used different 13C tracers to look at how different carbon derivatives can go into the intracellular metabolism. And that's a way to look at how the cells can preferentially use one carbon versus another. And that's very important because in real systems, you're going to have a mixture, not just one substrate. On this end, I just want to thank you for your attention, and I will be very happy to answer questions. I want to acknowledge my funding sources, which is NSF, the Department of Energy, and also the Center for Sustainability and Energy at Northwestern. Thank you.
Thank you so much. That was awesome. I have to brush up on my chemistry when I read your papers, but I really love it. There are questions in chat. Can you see chat?
Okay, let me go to chat.
If you could read a question out loud before answering, that's for the recording.
So we have a question from Sylvia. This is a very powerful technique. Did anyone ever do analysis of flux through cell-free systems? So we, as far as I know, let me think. So we have a collaboration with Neha Kamat, who is a biomedical engineer here, and she makes vesicles. And so we are in the process of developing a method to look at flux through synthetic vesicles. But I don't know if people have done it. I know people have done it to in vitro studies, just tracking a kinetic isotope flux profiling in vitro. But that's still something I think that needs to be done. So Wilson Bennett, did you ever think about looking through carbon conversion in the minimal JCVI? What does that mean? Cell?
That's the Craig Venter mycoplasma-based cell, the stuff that John Glass is doing.
Oh, okay. So this is a mammalian cell.
No, it's a mycoplasma.
Oh, it's a mycoplasma. Ah, okay. Did you ever think? I don't work with mycoplasma, but I think I would be interested in collaborating with someone interested in that. We have full computational. This is from the same person, Wilson. We have full computational model flux balance analysis for the SYN3A. It would be great to follow with experimental data. So I want to actually thank you for talking about flux balance. I wanted to highlight that the metabolic flux analysis that we do is different from flux balance analysis. So flux balance analysis is based on genome scale reconstruction and using biomass growth or growth rates as an objective function for biomass growth and using the gene. And this is why I went through the whole process of highlighting the difference between genome and the metabolomics. So what we do for our metabolic flux analysis is very high accuracy flux quantitation, because it is highly optimized on metabolite labeling at each node in your actual metabolic network. So it's not just saying that this is what I think genome scale reconstruction it's a powerful technique. I think it's helpful, but I just want to highlight that it's not for the purpose of looking at biochemical characterization at high accuracy is not high accurate, because as I highlighted throughout the talk, a lot of time you have the genes, but you don't actually have either the transcripts or even the proteins, or even worse, the metabolic fluxes that are associated with that. Okay, so Eva, sorry if I missed it. Could you do this metabolomics analysis on engineered metabolic pathways in vitro, just purified enzymes outside of the cell? So we don't need to do this because we do in vivo metabolic enzyme activity. However, you could, of course, you can just measure the metabolites. Yeah, you can measure the metabolites. I mean, you see the exometabolomics, we are measuring metabolites outside the cell. So you could do it. Let's say we had a collaborator who was just really interested in 3 metabolites. Of course, we have techniques that can look at 300 metabolites, but he was interested in 1 reaction. So you can do your, kinetics experiment in vitro, or purified enzymes, and so on. It's just that we don't take what's happening for our purpose. I'm not saying that to do it for your purpose, but for our purpose, we like to see what's happening in vivo, measure metabolite levels in vivo, and measure metabolic flux in vivo, because we know that's what's giving us the mechanistic information that we want. But we can for sure measure metabolite levels outside the cell. This is from Song Feng. Are there other possible regulations such as allosteric inhibitors, redox modification on proteins? Yes, that's part of the Nature Chemical Bio paper that I didn't get the chance to go into, but we did look at allosteric inhibitor and also allosteric enhancers as well. There's a whole figure on that. Thanks for that question. Taesuk, great talk. Glad to listen to your talk, although I'm very familiar. Actually, Taesuk wrote perspective views with Yin-Jae Tang for the paper. So thank you to Yunsu, and good to see it again, kind of in a presentation mode. So he said he's very familiar with the paper, the Nature Chem Bio paper, because he wrote that perspective. And I think this is the last question. Is there anyone else? I think, yeah.
I think that's all. I'm really glad that someone asked about the JCVI mycoplasma cell, because I was meaning to ask that.
Yes, yeah, we haven't done ourselves because we focus on bacteria, but we've had, I was actually talking to your colleague the other day. We have a couple of publications looking at the gut microbiome in insects and in ants, which is not what we do at all, but it's because of kind of the techniques that we use, is translatable to other systems as well. So we've done mammalian cells, we've done insect cells, we've done ant cells. So I'm sure that's even much simpler than these types of systems.
Yeah. All right. If there are no more questions, I want to say thank you so much again for a great talk.
Thanks, Kate.
And thanks, everyone.
Bye.
Bye.
Oh, there is someone. Oh, just someone saying thank you. Thank you, Maya. Bye.
Thank you. Bye.
I'm recruiting postdocs, by the way, if people are looking for postdocs positions. Okay, bye.